Hello everybody, my name is Juanita Costa and I will be talking about anthrax. So a few of the things that I will be discussing in this presentation include information on the infectious agent slash pathogen that causes anthrax, as well as signs, symptoms, diagnosis, and treatment of the disease. I will also be touching on two notable outbreaks of anthrax in the last century and a few reported cases that have occurred on American soil in the last 2 decades. So, what is anthrax? Anthrax is a non-contagious zoonotic disease caused by Bacillus anthracis. The organisms most susceptible to this disease are wild or domestic herbivore grazing animals, such as cows, sheep, goats, and horses. Susceptible populations include military personnel, wool sorters, veterinarians, farmers, and people who live in rural or agricultural areas. Um, Anthrax is most commonly seen in regions of sub-Saharan Africa, Central and South America, Central and Southwestern Asia, and Southern and Eastern Europe. However, they are not limited to these regions. They occur in the US as well, they occur in Canada, and they occur in other parts of Europe as well. Anthrax is also known as wool sorters disease, rag pickers disease, Siberian plague, and the black bane. Okay, so Bacillus anthracis is a facultative anaerobic gram-positive rod bacteria that exists in two stages, and these two stages are the vegetative cells and the dormant spores. So the vegetative cells are the growing and reproductive cells of Bacillus anthracis. The anthrax vegetative cells germinate and multiply inside the spleen and lymphatic system of the host animals. Then, after the animal dies, the vegetative cells are released into the environment, particularly the soil, which is exactly where these herbivores acquire the disease from, and those vegetative cells begin to sporulate due to the exposure to oxygen. These spores can be dormant for up to decades. So most strains of Bacillus anthracis are pathogenic except for the Stern strain, which is also known as 34F2 strain. This strain is a naturally occurring avirulent strain of Bacillus anthracis that lacks the PXO2 plasmid, and it was discovered in the 1930s. So what is the PXO2 plasmid? That leads us to the discussion of the two plasmids responsible for Bacillus anthracis' virulence, and these two are PXO1 and PXO2. These plasmids contain the genes that encode for anthrax, toxins, and a polydglutamate capsule, respectively. So PXO2 contains the genes for the polydglutamate capsule, and PXO1 contains genes for the anthrax toxin. So the polydglutamate capsule of Bacillus anthracis gives it an evolutionary advantage because it makes the cell antiphagocytic, and it also inhibits dendritic cell maturation in the host organism. So as I said before, PXO1 contains genes for anthrax toxins, which are these three here: lethal factor, also known as LF, protective antigen, PA, and edema factor, EF. So the combination of PA, EF, and LF causes fluid buildup in tissues such as the lungs and toxicity to host cells. EF is an enzyme which produces loss of water regulation in cells and a loss of ion metabolism as well. LF cleaves proteins from signal transduction pathways, causing a variety of effects, including cell death and interference with the immune response. So we've discussed Koch's postulates in class before, but I figured that it was worth mentioning since Koch did use and studied Bacillus anthracis to develop his postulates. A little reminder of how that went down. So he inoculated mice with the Bacillus anthracis taken from a diseased farm animal that died from anthrax, and he eventually began to isolate and grow Bacillus anthracis in pure culture, injecting these bacteria into mice. This led to the development of Koch's postulates, which again was important because it helped identify the causal relationship between specific microorganisms and disease. This is just a picture of two pictures of Bacillus anthracis. The one on the left is Bacillus anthracis, and this like white thing in the center, those are endospores, and the reason why they're white is because they're highly resistant to staining, and they are also highly refractile to light. And this is a scanning electron microscope image of the spores of Bacillus anthracis. Okay, so there are three main types of anthrax, but there's a fourth one that was recently developed or added, determined. But the main three are cutaneous, gastrointestinal, and inhalational anthrax. The fourth one is injection. So cutaneous anthrax. It develops after 1 or 7 days of exposure, and the incubation period is up to 17 days. Has been recorded to be up to 17 days. The fatality rate for this form of anthrax is somewhere around 2% with treatment. And the main signs and symptoms that you will see and experience with this form of anthrax is first, it begins as small blisters forming on the hands, face, arms, or neck, blisters or bumps, and these blisters get swelling around the perimeter, and they eventually develop into these blisters with a black center that will eventually peel off and just kind of flake or fall off. It's also worth noting that despite how gruesome they look, they're apparently not painless, and they're not itchy at all either. Additionally, 95%, over 95% of all cases of reported anthrax are cutaneous anthrax. And on injection anthrax, it is very similar to cutaneous anthrax, but it can spread through the body quicker because injected into the body, and sores and ulcers develop at the site of injection. So gastrointestinal anthrax. This develops after 1 to 7 days of exposure, after exposure, and the incubation period has been reported to be up to 16 days. The fatality rate for this one is much higher than cutaneous anthrax. It's somewhere around 40% with treatment, and without treatment, that goes up to 50%. If you have oropharyngeal anthrax, you will experience fever, chills, dysphagia, which is difficulty swallowing, and regional swelling of the lymph nodes. If you experience intestinal anthrax, you will experience diarrhea, nausea, vomiting, and GI bleeding, which can be seen in this image here. This is an animal who does not have anthrax, and this is an animal with anthrax, and this is that GI bleeding that I was discussing. Okay, so inhalational anthrax. This develops within 7 days of exposure, and the incubation period for this one can be up to 2 months, which is again due to those spores. The fatality rate with inhalational anthrax is up to 45% with treatment. The symptoms are fever, chills, fatigue, shortness of breath, profuse sweating, confusion, headaches, and It is worth noting that due to these symptoms, it's difficult to distinguish from influenza, COVID-19, or community-acquired pneumonia at times. The previous mortality rate for inhalational anthrax in 2001 was 90%, so it has drastically declined in the last 20 years. Um, and X-rays and CT scans are used when inhalational anthrax is suspected to check for pleural effusion, which is what you can see here, and mediastinal widening, which is what's going on here. Okay, diagnosis and treatment. So the only way that you can diagnose anthrax is if you do lab testing. So you can test for the bacterial DNA, toxin, or the antigen detection. You could also perform bacterial cultures of Bacillus anthracis. And again, you could do those imaging, the X-rays and CT scans when inhalational anthrax is suspected. So the main form of treatment for anthrax is antibiotics such as penicillin, which can be used to treat all forms of anthrax, clindamycin, ciprofloxacin, and doxycycline, and you can also use anthrax toxin as a treatment, but it's usually reserved for inhalational anthrax. Okay, outbreaks. So again, this is kind of a map to show y'all where anthrax is like most commonly happening. So here's that Central and South America, Sub-Saharan Africa, Southern and Eastern Europe, and then Central and Southwest Asia. But again, it can happen globally. Okay, so outbreaks. In 1979 through 1985, in Rhodesia, which is modern-day Zimbabwe, a study, so originally the numbers said that it was over 10,000 human cases and thousands of cattle cases, but with 200 human casualties. But a recent study revisited this outbreak and found that the human cases were actually well over 17,000, and the cattle cases were almost 200,000. And then for this last one, this happened in the USSR, so modern day Russia, in Sverdlovsk. And so there were 96 cases of human anthrax, where 17 were cutaneous and 79 were gastrointestinal. 64 of these cases ended up being fatal and people ended up passing away. So this occurred on Compound 19, which is a military compound that sought to create Bacillus anthracis on a mass scale. The outbreak was caused by an accident at the compound where air filters were not working properly, and they basically ended up accidentally releasing anthrax spores into the city. Okay, recent cases. So in 2001, there were 22 cases of anthrax, 11 were inhalational, and 5 people ended up passing away. So if none of you are familiar with that, after the 9/11 attacks, a government employee of the U.S. Army Research Institute for Infectious Diseases intentionally distributed letters full of anthrax spores through USPS. And that's how these people ended up getting anthrax. In 2006, a drum maker got inhalational anthrax from goat skins that he brought home from Africa to New York. And yeah, that's where he got it from. And in 2009, a woman contracted gastrointestinal anthrax because she had been participating in a drumming event. And the drums that she used were made from animal skins contaminated with Bacillus anthracis spores. And that is all. Thank you.